Hello, friends. Our today guest lecturer is Aubrey de Grey, essentially the most recognizable person in modern gerontology with an enormous following and quite a bit of clout. He doesn't need introduction. Still, I feel obligated to give some, at least. So he's BA in computer science, his PhD in genetics, and his alma mater is venerable Cambridge, I believe. I looked up his publications. It's well over 200. I just stopped counting, and several books. And he describes himself as of in a recent tweet, I'm spearheading the global crusade to defeat aging. So let's start with the lecture, and the floor is yours.
Well, thank you. Yes, so I am very grateful to you, Magomed, for having me on your podcast. I have definitely had great respect for many years for the rigor that you bring to thinking in this field, which I think we both agree is relatively lacking, and we could do with more of it. I am going, therefore, to speak today. I'm going to talk about, in a rather introductory way, about the way in which I look at aging and the various options for bringing it under medical control. And I'm going to talk about the ways in which I and others have discussed the feasibility of these various approaches. I think that there is still very much room for debate, room for reasonable people to agree to disagree on this question. And therefore, I think it's very important for people to understand where that debate actually sits and where it does not sit. In other words, what people are still on a range of opinions on, and what people, at least people who have thought about these things carefully, pretty much agree on. A lot of the reason I want to do that is not just to correct misconceptions in regard to what we might be able to do about aging in the foreseeable future, but also to focus people on priorities. In other words, to identify and highlight the areas in which people, both in the scientific community and other people within the wider gerontology community, can operate, can work to hasten the defeat of aging, and thereby, of course, to save innumerable lives and to alleviate an astronomical amount of suffering. All right, so with that little preamble, let me proceed. The way in which I think about aging and how we might go about fixing it is somewhat different from what has been traditionally thought, though I'm pleased to say that my way of looking at this appears to have gained a great deal of mainstream acceptance over the past decade or so. But I think that a good place to start in discussing all this is to start with a definition of what aging is. And this is something that I feel a lot of people, skip over to the detriment of what they say next. Now, of course, different people have different ways of defining aging, but this is one of the things where I think it's important to highlight that there's not really very much disagreement. It's just different ways of saying essentially the same thing. So I would like to claim that the definition of aging that I'm going to give you right now is, even though it's not necessarily the form of words that other colleagues might use, nevertheless, it's not controversial. It's not something that very many of our colleagues in the field would actually dispute. So here it is. It's a three-word definition, the three words at the bottom of this slide: metabolism, damage, pathology. All I'm saying here is that aging consists of a wide variety of self-inflicted, accumulating changes to the molecular and cellular structure of the body, which the body is set up to tolerate a certain amount of, but only a certain amount. And because those various changes accumulate without limit, that threshold tolerable amount is eventually exceeded. So we start to get sick, and then sicker, and then dead. And we can say that in 3 words. Metabolism is, of course, the word that biologists use to denote the entire network of processes that keep us alive from one day to the next. I'm using the word damage to denote those changes to the molecular and cellular structure that I mentioned. And the reason that it's appropriate to use the word damage is because of that threshold I just mentioned. The fact that only a certain amount of that change is manageable, is tolerable. And after that threshold has been exceeded, we get sick. And that, of course, is denoted here by the word pathology, the collection of chronic progressive health conditions characteristic of late life. So, of course, metabolism goes on throughout life, starting even when we're at conception. Because of this threshold, the emergence and progression of pathology does not really begin until late life. And so now that we have that definition of aging, we can, of course, dive in and say, well, okay, what could we do about it? And first of all, let's just remind ourselves what we mean by doing something about it. What it simply means in terms of this definition of aging is separating metabolism from pathology. In other words, allowing ourselves to continue performing metabolism, in other words, being alive, without eventually getting sick. And the original approach to this is what I'm calling here the geriatrics approach, to separate metabolism from pathology by separating damage from pathology. In other words, we kind of just fatalistically accept that metabolism is going to cause these changes, and we simply try to make the body more robust and able to tolerate greater amounts of damage before getting sick or before getting a given degree of sick. And, you know, well, I mean, a lot of effort has gone into that, and of course still does. A great deal of money is spent. In fact, the overwhelming majority of medical treatment for health-related problems of late life today consists of exactly that, trying to manage the damage and keep people as healthy as possible given the damage. But, of course, it's a losing battle. The more damage there is, the harder it is to prevent that damage from making us sick. Now, of course, I'm not the first person by any means to point this out. In fact, the whole field of gerontology really came into existence more than a century ago because a few people saw that the geriatrics approach to addressing the health problems of late life was a losing battle. And therefore they said, well, okay, we've got to be more preventative. We've got to intervene at an earlier stage in the chain of events. And the natural alternative to separating damage from pathology is, of course, to try to separate metabolism from damage. Fine. Thing is, it's not really fine because it's not possible. The idea here was inspired, of course, by the living world, by the fact that we see in nature a huge difference between different species in terms of the rate at which metabolism generates damage. And of course, this is, a source of great interest, and there was originally some degree of merit in the idea that we might be able to study this variation among species, and indeed even within species, and try to understand it. And eventually we might understand it well enough that we could extrapolate from that understanding to the devising of therapies that would slow down the rate at which human beings generate damage from their metabolism. In other words, essentially, to put it in terms of inanimate machines, let's say simply we make metabolism run more cleanly. Okay, but of course the difficulty here is that the creation of damage is so inextricably intertwined with the vast complexity of metabolism that really it's a non-starter. We cannot reasonably expect to be able to slow down by a significant amount the amount of damage, the rate at which damage is generated by our metabolism. And this wasn't very hard to really realize. There was one big complication, though, that held up the understanding of this fact. And that was the discovery that if you take rodents and you feed them less than they would like, they live quite a lot longer than they otherwise do. This was a discovery made around a century ago now. And of course, it is by far the most studied and most reliable way to increase the longevity of laboratory organisms. But it's a distraction because it is not really a case of messing with metabolism, of trying to make metabolism run more cleanly. It is a pre-programmed, built-in feature of metabolism. In nature, of course, the goal is to pass on one's genetic information, one's genes, and the availability of calories is unpredictable. There are famines in the real world. So it makes sense that that all species have learned to optimize their metabolism to respond to nutrient availability. And it turns out that a big part of what that means is that in a famine, you hunker down. You slow down the, you essentially reduce the rate at which you try to reproduce, which means that you reduce the rate at which you try to grow if you're still not an adult yet. And you use that energy, essentially, to ramp up the extent to which you maintain the body, which essentially means cleaning up let's call it pre-damage, cleaning up things that might become damaged. So you do indeed end up living longer as a result. The difficulty with all of this from the point of view of human beings, however, is that long famines are rare. Long famines are rarer than short famines. And that means that the evolutionary pressure to maintain the ability to respond to nutrient shortage is weaker in longer-lived species. And sure enough, we find that if you look at different species and you ask by what percentage can their longevity be increased using this method, using calorie restriction, we find that long-lived species get much less of a benefit than short-lived ones. And of course, we are a long-lived species, so we're rather out of luck in that regard. There is, of course, another difficulty with any method of what I'm calling the gerontology approach here, the slowing down of the creation of damage, which is that you'd better start early. If you take someone who's already in middle age, they've already accumulated at the normal rate most of the damage that is required for them to start getting sick, which means that even if you, let's say, were by some miracle to be able to halve the rate at which subsequent damage were created, you would still only rather modestly delay the point at which the total amount of damage reaches the pathogenic threshold. So all in all, it's a bit of a damp squib, I think is probably the word to say. It's not quite such a brain dead idea as the geriatrics approach, but it's really not very promising. And by the 1960s or 70s, the field of gerontology had more or less resigned themselves to this fact. And it had become very, politically incorrect to talk about doing anything about aging. Gerontology had become a bit like seismology. People who study earthquakes, they know very well that the thing they study is bad for you, but they have no aspiration to stopping earthquakes from happening. And it was a bit like that back then. And it stayed that way. Again, there was a bit of a distraction that happened in the late 80s, early 90s, when it was discovered that one could very dramatically increase the longevity of certain very short-lived laboratory organisms, initially nematode worms, of course, by simple genetic manipulation. This was, unfortunately, really just harking back to the calorie restriction intervention, because it was essentially tricking the body by genetic means into believing it was in a famine when it wasn't. And so, of course, it's simply emulating the response that is relatively ineffective in human beings in the first place. Now, of course, we have other methods, drugs and dietary interventions that also trick the body in this same way. We call them calorie restriction mimetics. And again, they're not completely useless by any means. They certainly do have the capacity to postpone the ill health of old age, especially in certain contexts where there is a challenge, for example, when someone is overeating. But it's not the Holy Grail. I came into the field 25 years ago or so, and I immediately decided, well, this just won't do. And luckily, I was able to come up with an alternative, which, of course, is center stage on this slide, the maintenance approach. Recall that we are all about separating metabolism from pathology. And what I've done so far is to disparage, I should say, the alternative of doing so by separating damage from pathology, and also the alternative of separating metabolism from damage. But we can separate metabolism from pathology in a third, completely different way, which is by repairing damage. If we can do that, then we don't need to interfere in either of the processes, the ones that cause damage or the ones that damage causes. We can let metabolism create damage at the natural rate, and yet if we are periodically repairing that damage, even if we're only repairing it imperfectly, we will be able to keep the overall amount of damage down below the threshold that causes us to get sick. So that is the approach that I've been pursuing ever since I realized that it was so promising back in the year 2000, 22 years ago. The reason it is promising, of course, is because it is something that can be fleshed out into actual concrete strategies. So here is simply a summary of what I've said so far. I rather like to highlight this kind of soundbite at the top, the fact that we can use the exact same definition of aging if we're talking about aging of a car or an airplane or any other simple man-made machine. It's not the fact that the human body is alive that makes this a good definition. It's just a feature of physics that any machine with moving parts is going to do itself damage, which is eventually going to stop it from working so well. So let us talk about the complexity of damage. Now, this is the real kind of, I don't know, the breakthrough I made in the year 2000, realizing that even though there are many, many, many different types of damage that we have accumulating in the body during life, and of course, I'll talk more about that in due course, nevertheless, it appeared to me that we could actually classify all of those types of damage into a relatively manageable number of categories, just 7 categories, which I'm listing on the left-hand side of this table. And back then I thought to myself, well, okay, how confident can I be that this is an exhaustive classification? And I thought, well, okay, the best way to ask that question back then was, for how long has it been, this list? And the answer was 18 years. The most recent example of anything being added to that list had been in 1982, when Richard Cutler came up with what he called the disdifferentiation theory of aging, which we now think of as essentially the accumulation of epimutations, changes to the epigenetic state, spontaneous, adventitious changes to the epigenetic state of a cell. And the year before that was when Monnier and Sarami came up with the idea that non-enzymatic glycosylation, and especially cross-linking caused by such a method, by such mechanisms, could impair the elasticity of the extracellular matrix. And that, of course, has also become a very important theme in gerontology. But nothing since then. I thought, well, that's pretty good. Now, of course, we have to acknowledge that until I started thinking and talking this way, people had not been trying to come up with such a classification. So maybe I was being a little unfair in being optimistic about this, because, you know, maybe other people would, once they started thinking about it this way, they would say, oh no, here is category number 8 and so on. But I've been saying this, I've been making a hell of a nuisance of myself for 20 years now, talking about all this, and I still seem to be getting away with it. It still seems that there is no need to extend this list, and that's really good news. So, of course, that's only part of the good news. The really big part of the good news is the right-hand side of this table, the fact that we have very practicable ways to do the actual damage repair, the maintenance approach in each of these cases. And this is really the value of this particular classification. The classification is precisely designed so that for each category, there is a corresponding generic approach to doing the repair. So just for illustration, let me just talk about the first one. The point here, of course, is that stem cell therapy, that's what it is, right? The idea is when you have cells dying and not being automatically replaced by the division and differentiation of other cells, then the number of those cells goes down, the cellularity decreases, and eventually that's bad for you. So we can look at, for example, Parkinson's disease as a classic example of this, where the dopaminergic neurons in the substantia nigra are very highly energetic. They damage themselves rather more rapidly than your typical neuron does in the brain. And sure enough, they die at a more rapid rate than other neurons. By old age, we have pretty much all of us lost maybe a quarter of the dopaminergic neurons that we had in youth. And that's okay, there's that much margin of error in the system. But of course, as with anything in aging, different people experience different types of damage at different rates. And sure enough, some people by old age have lost maybe 3/4 of their dopaminergic neurons, and that is too much. And that's why they have the symptoms of Parkinson's disease. So the question, of course, then is, what do we do about it? And the damage repair is to replace the cells. And that's what stem cell therapy is. You prepare cells in the laboratory to into the right state so that they know what to do to go back into it, so that you can put them into the required region, and they can divide and differentiate to replace the cells that the body is not replacing on its own. Sure enough, it seems to work. Back in the 1990s, people tried this for Parkinson's disease in Sweden, and mostly it didn't work. Mostly there was no benefit. And we know why that was. It's simply because the amount that we knew back then about how to manipulate stem cells in the laboratory was essentially zero. And so all that could be done was to isolate cells from roughly the right part of the brain of aborted fetuses and to inject those just unmanipulated into the brain of Parkinson's patients. So, of course, by and large, people didn't respond because they didn't get lucky. They didn't get any of the right kind of stem cell. But a small number of patients did respond because they did get lucky, and the response was spectacular. There was a little case report written by the group about 5 years ago or so, which was essentially a case study of the first patient who actually did respond. And essentially, you know, they were able to be taken off L-DOPA a year or two later. They didn't, even then, with just one stem cell injection, they didn't get any Parkinsonian symptoms at all again until maybe 10 years later. And even then, they only came back very slowly. So an extraordinarily effective treatment. And now that, of course, we have induced pluripotency and all that, people are very optimistic that this kind of treatment is worth having another go at. And sure enough, there's a clinical trial in Japan already underway, and there are a couple of other clinical trials in the US just about to start. So everyone's very hopeful. Now, first of all, let me not give you the impression that I don't know that this is not the only use of stem cells. Certainly, allogeneic stem cells are being used in many clinics already, simply because even though they're allogeneic and after differentiation at least, they become immunogenic and therefore are removed by the immune system, nevertheless, in the meantime, they secrete good stuff. And so we appear to have rejuvenating effects from stem cells even over and above cell replacement. But let me just focus here on cell replacement and highlight the fact that, you know, different tissues have obviously different types of stem cell, and therefore different stem cell therapies have differences. Therefore, I am not simply saying we will have a one-size-fits-all treatment for all cell loss, not at all. But we must not take that concern too far either. We must remember that even though different stem cell therapies have definite differences, and that's why there's an entire field called stem cell research, nevertheless, they also have an enormous amount in common, which is why it's one field and not just a disparate bunch of, you know, different groups, some working on one stem cell and some on another. And exactly the same kind of concept applies throughout this list. I won't go into all the details right now for lack of time, but I just want to emphasize that this is the purpose of this classification. And that's kind of in contrast to the classification that has had the largest amount of exposure and attention over the past decade. This is, as I guess pretty much everyone in the audience will know, a couple of figures from a paper that came out in 2013 called The Hallmarks of Aging. And that paper was very good news for me, even though they didn't cite me, because the fact is, it was a restatement of what I had written and said more than a decade earlier. It was to highlight and to point out the feasibility of a divide and conquer approach, a divide and conquer damage repair approach, in fact, to the various types of damage that accumulate during life. As you can see, and as you almost certainly already know, these authors chose to divide the phenomenon of aging into 9 different categories rather than 7. But that was not because they identified things that I'd overlooked. It was simply a different way of dividing stuff. In fact, they actually overlooked things that I had not overlooked. They didn't have anything about glycation in there, which was a bit of an embarrassment. But really, that's not the main difference between their classification and my original one. The main difference is that this classification was not designed to, to correspond to damage repair therapies. So the discussion of what damage is and what the damage repair things was a bit more, well, I would say a bit more convoluted. I still prefer my description. But of course, what really matters here is that when I came along, I was very much ahead of my time back in 2002 when I published all of this, even though I published with some very distinguished co-authors. Whereas by this time, the dust had kind of settled. I had kind of won the argument. And so this paper became by far the most highly cited paper in the whole of the biology of aging last decade. And ever since it came out, I have never had to justify the damage repair approach to other gerontologists ever again. So that's really rather heartening. However, there's more, because even though I may so far have sounded very bullish about all this is where I want to come to the really important discussion of how hard the damage repair approach really is and really will be to implement. And I have discussed this over the years with a number of people, including you, Magomed, of course. But I do want to highlight a discussion that I was able to persuade my great friend Vadim Gladyshev from Harvard to engage in with me on stage at the conference I hosted in Berlin in 2019. I believe that the video of this little debate is, is still online, so I encourage you to go and look for it. But I asked Vadim to do this because he is, he thinks very similarly to me in terms of how to describe aging, how to go about fixing it. But he's considerably more pessimistic than me when it comes to how difficult it's going to be to actually get the whole thing to work. And he has written various very powerful reviews and publications on this topic. So again, I encourage you to look those up. But the essence of it goes like this. I say these things. And, it's like, I've highlighted some of this already, but let me go through them. The way in which the damage repair approach was widely misunderstood back in the day when I was first propounding it was that to many gerontologists, it sounded indistinguishable from the geriatrics approach. Problem with the geriatrics approach I explained earlier is, of course, that it's a losing battle, that as damage accumulates, it becomes harder and harder to stop it from becoming pathogenic. But there's a second difficulty with the geriatrics approach, which is that all of these types of damage become pathogenic at more or less the same age. And this, of course, is very much what one would expect from evolutionary theory. There's no point in having different, in having different types of damage, some types of damage be, if you like, unnecessarily well protected against, because they will simply, that extra protection won't confer any benefit. if the individual is dying of something else. Hence, that unnecessarily good protection will progressively degrade in quality, and you'll end up with everything happening at roughly the same age. But these considerations, it seems to me, are easy to get overly worried about because of a variety of different things that allow us to essentially have economies of scale and to kill multiple birds with one stone and so on. So I'm going to talk about those in a little more detail. Let me remind myself what we've got here. Oh yes, I'm going to try and get rid of this. Sorry, hang on. Yeah, I'm having a little difficulty because I can't see the top of the titles of my slides because it's covered by a Zoom menu, but I'm coping. Yes, there are lots of different types of damage, and we are discovering new ones all the time. But the good thing is we are not discovering, as I mentioned earlier, we are not discovering new categories of damage. The new types of damage we discover are, for example, new types of waste product that accumulate inside cells or outside cells. These are things for which the repair strategy is exactly the same as it is for the corresponding things in those categories that we already knew about. And therefore, it seems highly likely that we will be able to repair those types of waste product, for example, to develop the methods to do so very much more quickly, having already developed methods to remove one or two of them. It's just like reusing most of the knowledge that we already had. Right, again, I'm going to, as I've said, this is a problem that the damage will only accumulate at a rate that the body lets it accumulate, but how fast that is depends on how good quality, so to speak, the damage repair and maintenance machinery that we have naturally is selected to be. So what's the but? Why am I optimistic? Well, so this is something I've alluded to already, but let me emphasize it further. This clustering types of damage into categories is not just for comprehensibility. It's based on these generic therapies. I went, of course, into the case of stem cell therapies in some detail. And yeah, so I mean, really, you know, think about it. If you've got, for example, an immune therapy to cause macrophages or microglia to engulf a particular type of amyloid in, for example, the brain. We already have, of course, therapies that do that. They don't have much clinical benefit against Alzheimer's because, of course, Alzheimer's is a very multifactorial phenomenon, but they do get rid of amyloid. They chop it up and destroy it. And we have pursued work to essentially emulate that approach against other amyloids, in particular transthyretin amyloid, which accumulates in the heart and certainly causes a rather nasty type of senile systemic amyloidosis. And it wasn't very difficult. You know, the group that we funded was able to repurpose their technology to target transthyretin, and we were able to publish several years ago now a very promising proof of concept of that, which has now been sufficient to actually allow the researchers in question to quit their jobs and go and start a company to take it forward faster with the larger checks that investors tend to be able to write. So this is just one example, but I really feel that it's a reasonable justification for expecting that as time goes on, our ability to knock off more and more types of damage will accelerate. Then, of course, yes, the question about natural selection causing damage, our built-in damage repair and maintenance mechanisms to be only as good as they are, actually is a little oversimplified. There are two ways in which that may actually not necessarily result in new types of damage coming along when we're only slightly older than the types of damage that we already know about come along. First of all, there may be types of damage which just accumulate so slowly that they don't matter at all. And the main example of this that seems to be an example anyway, is racemization of amino acids, especially aspartate, in the extracellular matrix. So racemization, the switching of chirality of amino acids in a protein, is something that could potentially be immunogenic. It could potentially alter other aspects of the behavior of the protein, if it's an enzyme, for example. And sure enough, inside cells, intracellularly, there are mechanisms to eliminate or to reverse racemization of aspartate. And these have been genetically characterized, and mouse mutants have been made and they're not happy. But it turns out that the rate of racemization is dependent on a number of things, one of which seems to be a very important characteristic is the rigidity of the protein backbone, which of course is, it's pretty rigid in the extracellular matrix. So sure enough, aspartate racemization outside the cell is rare and it's slow. And it may be that we just don't have any system to fix it because there's so little of it, even by old age, that evolution just has had no reason to care. And it won't make us sick until we get to be a lot older than we are now. We could be wrong about that. We might have to fix it. But as I say, we've already got enzymes that fix it inside the cell, so maybe that won't be very hard anyway. There's another thing, though, which is something that I call protagonistic pleiotropy. And let me just check whether I have a slide on this. I don't. Yeah. So yes, I wrote a paper called Protagonistic Pleiotropy some years ago, and I focused in that paper on cancer, or more particularly on mutations in the nuclear genome. Essentially, I said, well, okay, all of our nuclear genome is maintained and protected by the same machinery. In other words, genes whose inactivation might predispose to cancer, or whose overactivation might predispose to cancer, are protected by exactly the same machinery as genes that have nothing to do with the cell cycle. And that's rather important because it means that the requirement not to die of cancer before you have had offspring has driven evolution to create a DNA repair and maintenance machinery that is good enough to slow down cancer to that extent. And because that same machinery is used elsewhere, that means that it ends up being unnecessarily good, because the problem with cancer, of course, is that just one cell going really wrong can kill you. Now, of course, there are plenty of other mechanisms against cancer that happen once the cancer cell starts to show that it's gone wrong, but those are only partially compensating for the problem I just mentioned. So that means it looks, at least to me, as though we do indeed have only a rate of accumulation of genetic damage in the nucleus that is independent of the cell cycle, that is far too slow to make us sick in anything like a currently normal lifetime. I believe the best evidence for this comes from the group of Jan Weich, now at Albert Einstein College of Medicine, previously at the Buck Institute, and previously in San Antonio. He, over many years, looked at, used various models of to measure mouse nuclear genome mutation accumulation, and in my view showed very convincingly that in most tissues the rate of accumulation during adulthood is so negligible that it is very hard to blame anything on it. We actually funded Jan's group for some years to look at the corresponding question in relation to epimutation, so spontaneous changes to the epigenetic state, especially to the methylation state of cells. That was a very challenging project back then, because back then there was no good way to to do bisulfite sequencing, the way that you distinguish between cytosine and methylcytosine at a single cell level. You needed thousands of cells, which precluded the measurements that we wanted to make. And the fantastic postdoc that we were able to fund in that group essentially solved that problem, or at least to a large extent. But we still don't have a firm answer to this question, but preliminary indications are that indeed the situation with epimutations is the same as with regular mutations. Very important also to understand what we mean by damage in relation to DNA, because unfortunately there is a terminological confusion. Specifically, people who work in DNA repair and maintenance use the word damage to describe something completely different from what I use it to mean. What they use it to mean is pre-mutagenic lesions, things like 8-hydroxydeoxyguanine or mismatches, things like that. Things that are amenable to repair by enzymatic machinery that we already have in the body, and that, of course, by and large, are indeed repaired before maturing into a more stable form. And that's fine. So when they do mature all the way to actual mutations, then people who work in DNA damage and repair, they don't call them damage anymore. Whereas those are the things that I do call damage. So this is a terminological, inconvenience. And I want to make sure that nobody misunderstands that. Nevertheless, point is that when we measure the kinds of damage that happen, and this is not just DNA damage, those intermediates tend to be the things that we measure. And that can lead us to an incorrect conclusion with regard to whether we are actually repairing damage. In other words, if we are, for example, able to improve the performance of enzymes that repair pre-mutagenic lesions, then we will see a reduction in the abundance of those pre-mutagenic lesions. But we will not be doing anything to reduce the amount of actual mutations. And furthermore, we may, depending on the method, not actually be appreciably slowing down the rate of accumulation of those mutations. So, this can be highly misleading. Essentially, if we were able to do that, then geriatrics would actually work. So now, what would we do in terms of encouraging people to work on these things? Of course, I have very much not simply a curiosity-driven interest in this way of looking at aging and how to fix it. I actually want it to be fixed as quickly as possible. And that often means ensuring that people who are placed to do the work on it are able to get funded to move faster than they otherwise might. Now, one convenient feature of aging that is relevant here is that each category of damage that I enumerate in sense, except probably, really mitochondrial mutations, is instrumental, is primary in driving at least one major type of age-related pathology. Mitochondrial mutations, it's not too bad, but we're really looking more at orphan diseases of childhood that are driven by inherited mutations. But it's looking as though the others anyway, are benefiting from the fact that there is a particular pathology, a high-profile pathology that should be addressable by fixing just one type of damage. This is most convenient because the type of damage that you can fix, you prefer to be able to just focus on one type of damage and see a result that can actually confer benefit to people and therefore make money, even if on its own, it does not actually extend life because there are types of damage that it does not address and types of pathology that it does not delay. Plus, crosstalk This is a really good thing about the past few years, when the Mayo Clinic and the Buck Institute in their collaboration showed, starting about a decade ago, that removing senescent cells in a genetically engineered mouse was good for them. They were able to show that the mice benefited in a considerably wider range of ways than what one might have anticipated, implying that when you remove senescent cells, you are also slowing down significantly the rate of accumulation of other types of damage, or perhaps just like taking the pressure off other types of damage, automatic damage repair, and thereby having a much more wide-ranging effect than might have been expected. This is very encouraging. It says that we may be able to have a big effect, even in humans, on how, on the age at which people get sick, when our panel of interventions, our portfolio of damage repair, is considerably more incomplete than I would have guessed 15 years ago. But now let me get on. That's a kind of bridge to what I want to say in the last 10 minutes or so. I want to talk about the main challenges that we have to worry about starting now, which have been changing considerably over the years. They've been changing partly because starting maybe 5 or 6 years ago, we started to see investors coming in specifically focused on doing something about aging. Initially, this was typically angel investors, seed investors writing 6-digit, 7-digit checks to get startup companies going. And now it's definitely burgeoning into, more established, more traditional, more mainstream venture funding and so on. That's one thing. Another thing that's happened, especially during 2021, has been an explosion in the amount of nonprofit funding in the field, much of it coming from big hitters in the cryptocurrency community. And that's changed a lot of things for Sense Research Foundation, among other groups. But let me go through the challenges. First of all, short-termism. We know, of course, that anyone who wants to make money wants to make it quickly. And of course, that is true of the investors in the longevity field, in the longevity industry, just as it is true of investors in any other field. And therefore, companies that receive money from investors tend to have to worry about being pressured into focusing on the low-hanging fruit, on focusing on the easier areas. And similarly, in academia, we know that, it's all publish or perish. The more you publish, the higher your chances of getting promoted or getting your next grant application funded and so on. So for a different reason, there is just as strong a pressure on academics as there is in industry to focus on the low-hanging fruits. And so that's a shame, because the low-hanging fruit is often overpopulated by advances that will never scale, that will never actually have much chance of working particularly well in human beings, which is a great shame. And this has also sucked money out of the nonprofit sector. So Sense Research Foundation, as an independent nonprofit charity, has relied very much on people who in the rest of their lives tend to be investors rather than donors. And now that they can be investors in the longevity field, it has proven a challenge to maintain their interest in continuing to donate as well. Another one, combining therapies is boring. Yeah, I mean, supposing you've got two therapies that both extend the health span or lifespan of mice by some amount, and that's already been published, and you put the therapies together, and sure enough, they synergize. They extend lifespan by a bit more. Well, that's good. It's very good. It's very important. But it's not very publishable. It's not very interesting. You won't get published in Science or Nature or Cell that way. It's not eye-catching. Furthermore, it doesn't always work. Combination therapies very often interact unpredictably. And maybe this is less true in the case of damage repair. There's less inherent likelihood of such interactions, but it's still a thing. And so again, neither industry nor academia is incentivized for this. And for that reason, Sense Research Foundation is very strongly committed to expanding and spearheading over the next few years a big push to bring therapies together, to combine them in mice and optimize that combining process, because other people are just not doing it. Yes, so this is another point. So I was mentioning a moment ago that investors are very keen on low-hanging fruit. But some investors, in fact, especially some of the early investors, are also mission-focused to a large degree and are willing to cut some slack there and make sure that the companies they're investing in are doing work that will scale in due course. However, as a company grows, it brings in more investors. And unfortunately, that means it brings in investors that are less mission-focused, that are more focused on making money and less interested in how. And this certainly becomes very true at the point where a company goes public, or indeed gets acquired by a larger company. These, there have been some really quite, horror stories already in the industry in this regard, and it's going to be difficult to stop this from happening. But it's really something that you need to be careful about. I believe that founders of companies in this field who actually want to make a big contribution long term, as well as just making money for their investors and so on, and for themselves, these people need to be very careful to take the right money, the right kind of money at the early stage in order to be protected against being essentially diverted into being, in the long run, essentially a waste of time. Let me talk briefly about the Tame trial, the famous trial for the impact of metformin on human beings in terms of aging. Metformin, of course, a drug that's been around and used for diabetes since before I was born. Nevertheless, there is a recent, over the past decade or so, level of quite strong enthusiasm that the data may show in a tentative way that metformin is good for a wide variety of different aspects of aging. Of course, the reason why Tame is famous is because of the regulatory breakthrough, the fact that Neil Barzilai and other colleagues, after an extremely long and painstaking discussion with the FDA, were able to get the FDA to say, right, well, this clinical endpoint that you have devised is acceptable to us. If your trial succeeds in showing that it meets this clinical endpoint, then we will approve it for a wide range of applications, of conditions. And of course, the clinical endpoint was aging. Now, it was not described as aging. It was described in a very convoluted and complicated and combinatorial manner, but that was kind of the point. The idea was to incorporate many different types of clinical endpoint, each of which individually was already uncontroversial from the FDA's point of view, and to combine them in a way that incorporated the understanding of this crosstalk I was emphasizing a slide or two ago. Now, Tame has had a rocky ride when it comes to getting funding. Various people said they would do it and then didn't. In particular, the NIH ended up torpedoing the idea of funding half of it because the, well, there was let's just say that there was an enormous fight within the NIA, which led to certain resignations. But at least it now looks as though this is happening this year. However, the delay has definitely delayed recognition of how significant the Tame trial is, whether or not the Tame trial actually succeeds in meeting its clinical endpoint. And that will have delayed the impact that the Tame trial can have on the incentive structure and the decision-making process in the private sector, especially in big pharma. So that's a shame. But, if we focus on it, then we might be able to catch up. Finally, I just want to talk about policy. This is a really big thing now, because at the end of the day, governments have more money than anybody else, and they also have more clout in terms of responding to and indeed influencing public opinion. And policymakers around the world have, of course, had a very major topic of concern over the past couple of years, namely COVID, which, as we all know, hits the elderly very preferentially, even more preferentially than your typical infection. Therefore, of course, the priority, the importance of trying to rejuvenate and make more youthful the immune systems of the elderly is extraordinarily high now. we've really got to try and do it. And that means damage repair, of course, just like any other type of rejuvenation. Furthermore, there are already really good clinical trials, even small ones, in immune rejuvenation at this point. So throwing a lot more money, especially public money, at immune rejuvenation seems like a very good idea. We have to justify that. We have to make sure that governments understand that this is not money down the drain. And in order to make that happen, we have to understand why we have historically failed to get policymakers to understand the potential economic benefits of spending money on biomedical gerontology. The efforts to try and cause policymakers to understand that have been going on for a very long time, as I'm sure all of you know. Best part of 20 years ago, some extremely distinguished gerontologists, including the founding head of the NIA, Bob Butler, also J.L. Shansky, also a very prominent policymaker named Dan Perry, and also a very prominent and influential biogerontologist named Rich Miller. They got together and they started this thing called the Longevity Dividend Initiative, saying, listen, even a 7-year increase in health span would save the most insane amount of money. We'd like just 1% of that insane amount to be spent on longevity, please. And it made no difference at all. They had zero impact. And of course, the reason they had zero impact is because policymakers simply said, yeah, right, aging is like the weather. There's nothing that can ever be done about it. You just want more money for your labs, go away. Now, that cynicism, that skepticism is much harder to maintain, much easier to overcome, given the progress that's been made in people's laboratories and even in the clinic in actually progressing towards the medical control of aging. I want to highlight, just in closing, this guy, Andrew Scott. He's a British economist, and he last year wrote what I believe will be a very influential paper, really the first of a whole slew of papers that will be coming out of his research program, talking about, well, essentially reviving the longevity dividend idea, but doing so in a modern way, and especially talking about rejuvenation, talking about actual damage repair, as opposed to simply slowing aging down, which is what everyone had focused on beforehand. He even came up with a good name for the rejuvenation approach. He called it the Wolverine scenario. So yes, I am a massive fan of Andrew Scott, and I believe that his initiative has the potential to make a really big impact on the thinking that goes on in the corridors of power around the world. So it's coming up to the hour, so I think I will stop there. Magomed, if you have any brief questions, I'm happy to answer them, but I hope that's been entertaining.
Well, it was an interesting talk in many dimensions. Well, you showed gerontology is full of intrigue and suspense, which is good. But as far as funding is concerned, science in general is not supposed to produce any benefits. I mean, any profits. It's a long-term affair, which will hopefully generate something beautiful sometime in the future. And those who pour money into science, they do understand they cannot expect immediate returns. Moreover, they should expect lots of failures because before anything tangible, useful appears, right?
Oh, yes, definitely.
And in my opinion, to these principles corresponds the idea behind the Calico company, because they have unlimited resources and they claim to be strategic in their approach to solving the problem of aging. Well, I have a few questions to you.
Before you go on, let me say a few words in response to that, because I think you're making a very important point here. Really, when I came into the field, I wondered what to call myself. And it didn't take me very long to understand that I should call myself a biomedical gerontologist. This is a phrase that was first introduced by Denham Harmon. And the point is, it distinguishes me from, on the one hand, clinical gerontologists, geriatricians who are focused on doing the best they can with therapies that already exist. But it also distinguishes me from curiosity-driven biogerontologists who are focused on understanding aging for the sake of understanding it, and who do not really view the application of what they may discover as their problem. For me, I've always been goal-directed, but I've also been focused on doing the pioneering stuff that, as you say, is likely to fail very much more often than it succeeds. So the thing is, I believe that even now, as the private sector has become involved, investors have become involved, we need to maintain and highlight that pathway, that pipeline, if you like, between the concept and the clinic. And the good news is that investors see it that way as well. Investors understand that, at the early stage, things are very high risk. They're going to lose most of the time. They have to write small checks and, essentially hedge their bets by taking a lot of shots on goal, as some of them say. And luckily, they're willing to do that. So I think, we have to make sure that no gaps start to arise. In tech, in information technology, the phrase "the valley of death" is often used to describe all this, and also in biotech. And I've been very watchful in regard to the potential emergence of valleys of death in the pipeline from concept to clinic, and trying to stop them from occurring.
You're being less poetic than the Bible, because it's valley of the shadow of death. It's beautiful. But anyways, yes, what I'm concerned about, and you referred to it, that in my opinion, many primary mechanisms of aging, including telomere attrition, accumulation of senescent cells, mutation accumulation, you name it, or corrosion of epigenetic control, they're relatively synchronized.
Right.
So, or in other words, they reach critical threshold approximately at the same time, which essentially deprive any single mechanism being manipulated from ever affecting the general process of aging.
Well, I don't, of course, quite agree with that, because of this concept of crosstalk that I mentioned, and the fact that, especially in the recent years' experience of removal of senescent cells in model organisms, we've seen a rather more wide-ranging set of benefits than one would predict from viewing the various types of damage as accumulating independently of each other and causing pathologies independently of each other.
Well, I have to note here that somehow all the people who run the experiments on removal of senescent cells carefully avoided the primary theme here, in my opinion, that about 50% of brain cells have markers of senescence or show markers of senescence.
Mm-hmm.
So the primary benefit here in early experiments was that the senolytic employed didn't penetrate blood-brain barrier.
Could I stop you there for a second? Because of course the original work that got everyone so excited was not senolytics. It was the genetically engineered mice with the ability to simply knock out senescent cells using tamoxifen or whatever.
Yeah.
Now, if I'm not mistaken, those mice did have loss of senescent cells in the brain as well.
Yes, and here comes my question though. So if the primary purpose of any longevity strategy is to preserve our precious brain,
mm-hmm.
and irreplaceable cells mostly,
mm-hmm.
how do we go about it?
Well,
and what's the potential of this approach if it concerns only health span of the body apart from the brain?
So, of course, you're completely right that the brain is the organ that matters the most. But honestly, I don't think that we currently have data showing that we have a problem with that. The removal of senescent cells certainly needs to be done right. But if we ask, which cells in the brain show senescence, I'm not sure that we even have good data yet on the question of whether these are predominantly glia or neurons. And I would certainly start from the expectation that it's mostly glia, because
so you're placing your bet on uncertainty.
Oh,
Okay.
not at all. Not at all. It's a high profile thing. It's a very high priority thing to find out, but the lack of detrimental effects on cognition in mice in which senescent cells should have been removed, whether by genetic means or by pharmacological means, it kind of points in the direction of it's probably not anything that matters too much.
Well, that's exactly my point in a sense, because similar experiments with similar concerns concerning epigenetic rejuvenation.
Mm-hmm.
And they tested the animals after the treatment for formation of memories and so on and so forth, cognitive abilities and so on. And they showed that cognitive abilities do improve. But none of them showed that long-term memories were retained before the treatment, acquired before the treatment.
So could you possibly elaborate, Magomed, what study are you describing here?
I don't have anything with me right now to point exactly. Ocampo, right, 2016, something along those lines.
Okay,
And
well, what was the method of epigenetic rejuvenation?
it was OSKM, 2 days
okay.
a week, that kind of stuff.
No, I'm with you. Okay, so not actual epigenetic. Partial.
Well, what I'm trying to tell that previously acquired long-term memories were not tested in these mice.
Mm-hmm.
And I think that kind of research is crucial just to prove usefulness of this.
Let me comment briefly. So I actually think that we are going to need radically different ways to do partial reprogramming if we're ever going to make it useful in vivo. My main concern is not so much the one that you just highlighted, the possibility that a partially reprogrammed neuron might, lose information, but rather the oncogenic risk, the fact that, unlike a cell culture dish, the body is full of a rather large number of cells that are most of the way to becoming cancerous already. Hitting them with a sledgehammer like OKSM, even transiently, seems to me like a very bad idea.
Yes, that's my second consideration, yes.
Yeah, I am much more interested in options for partial reprogramming that simply do not have the capacity to take cells all the way back to pluripotency, but instead take them back only through the embryonic fetal transition, which is a much later event, and therefore restore the ability to regenerate rather than the fibrotic phenotype that happens subsequently for scarring and so on. I believe that has genuine potential. And one of the greatest tragedies right now is that the company which has been pursuing that and spearheading the possibility, which is Ajax, a company I used to work part-time for, is now in pretty much suspended animation through lack of funding. So I'm trying to fix that.
Mm-hmm. Well, there are many points you made, so I'm getting lost in all my little notes here because there's so little time to process them. Well, you were referring to obesity as the only proven method, but obesity is self-inflicted as well, right? So because
the only proven method of what? The only proven method of what?
Of radically extending lifespan.
Oh, you mean calorie restriction? Okay, right.
Yes, caloric restriction. But essentially, it's a self-created problem because all our pets, all experimental animals tend to get obese. All controls were ad libitum. So essentially, we're talking about metabolic correction of already pathological state.
I'm with you completely. I think that, in fact, it's even quite dangerous to call type 2 diabetes an aspect of aging because, strict control of diet is a very good way to very largely reverse it, even if it's relatively advanced. So I'm completely with you there. But the point is, even if we consider a population that is not obese, but is eating relatively ad libitum, so to speak, then still we anticipate, and we can indeed see in various experiments that there is this very steep inverse correlation between the natural longevity of a species and the percent benefit that it gets from calorie restriction. You know, the monkey studies, even the one that from Wisconsin that supposedly got a reasonable result, you know, the percentage improvement was very great, still less than you typically see in rodents.
Sure.
And that's despite the fact that, of course, there were concerns about the controls having an overly problematic diet.
So, and my concern is that all the methods you mentioned so far hit a roadblock with our individuality. And I don't see it surmountable at present, or at least all of these approaches should be tested against this dangerous possibility.
I completely think that, like any clinical trial, one should try to have as few exclusion criteria as possible. One should try to, you know, to manage heterogeneity of the population. However, this is something I probably should have highlighted earlier. Damage repair is much less vulnerable to a to a clinical trial working when it won't work in the real world as a result of lack of heterogeneity of the treatment group. The reason it is because we all accumulate the same types of damage. The only reason why some of us die of cancer and some of us die of heart disease and so on is because of rather modest differences in the relative rates of accumulation of different types of damage. And that's a much more, I would say, insignificant type of heterogeneity than what we see in people's actual metabolism, in, like, you know, people's, you know, ability to metabolize alcohol or sugar or whatever.
Well, I don't deny the value of your approach. It's essentially undeniable.
I wouldn't have come on your podcast if I thought you did.
Well, it's pure common sense, biological common sense.
Yeah.
But at the same time, the emphasis on post factum bothers me a little bit, because some losses are irretrievable. As you mentioned, that we're losing 25% of our brain cells, neurons.
Well, no,
and you cannot repair that.
No. We're only losing 25% of our dopaminergic neurons, which don't store information in the first place. So that's okay.
Okay. But some things cannot be solved. You can solve the problem of Parkinson's because it's a generic function neurons. So you put neurons there, and conceivably each human being should be cured completely.
Of Parkinson's, yeah.
Yes, but not so about other conditions.
Well, right. So one of the projects, so you're that's a great invitation to me to highlight one of the projects we're funding at the moment. So we're very interested, of course, in Alzheimer's disease. And while we do understand that eventually you become like Ronald Reagan and you've genuinely got a huge amount of loss of information, nevertheless, things can get pretty bad and still be recoverable. A lot of players made by Alzheimer's researchers about the fact that the hippocampus is particularly badly affected. And of course, the hippocampus does not store information, does not store long-term memories. It's a kind of pipeline between the short-term and the long-term. So there may be a kind of locked-in syndrome going on there. We may actually have plenty of information that appears to have been lost from the behavior of the individual, but can in fact be retrieved. And so we don't want to give up on people, not until we really have to. And that means that we do have to look at stem cell therapies for Alzheimer's, not just for Parkinson's, which is tricky because in Alzheimer's disease, of course, cell loss, when it does happen, neuronal loss is rather more widespread, much more widespread across the brain. So difficulty there is that if you inject a bunch of stem cells, ICV, then, you know, because the cerebral spinal fluid is basically sticky, viscous, the cells just kind of don't move around. So we are funding a really cool project at Einstein College of Medicine at the lab of Jean Hébert, which involves essentially having engineered neuronal precursor cells, which start out having a phenotype of microglia. So microglia actively migrate in the brain wherever they're needed.
Yes.
So the idea is you inject the cells, they do their microglia thing, before anything else, and then you flip a switch because, of course, you've done some kind of, tamoxifen thing, and you basically turn them into neuronal precursor cells when they have already migrated, and they get to do what you want them to do.
Mm-hmm.
So, of course, ambitious project, but logical, right, and realistic. In fact, sufficiently realistic that it's already getting private funding as well as public. So this is the kind of thing where, being a bit creative, in fact, being very creative about looking for engineering solutions to damage repair can surprise you.
Yes, I don't want to tax more of your energy, considering that both of us are people of advanced age already. But anyways, I'm really, really grateful. It
Likewise.
was a very enlightening delivery and lecture, and I'm pretty sure that our viewers will also appreciate it.
Well, I'm very glad you enjoyed it. Thank you for having me on the show.
Thank you so much.
Bye-bye.
Bye.